Good day, everyone. My name is Nur Adina binti Zainal Abidin, and I am from the Master in Molecular Medicine ODL program in IMU. Today, as part of my Immunology and Infection module, I will be presenting my topic on the role of mRNA vaccination technology in the fight against emerging infectious diseases. This presentation will consist of the introduction to vaccination and immunization, literature reviews, introduction to mRNA vaccination technology and its advantages, case study trial with COVID-19 using mRNA vaccines, application of mRNA vaccination technology against emerging infectious diseases, its challenges and limitations, as well as future prospects of the mRNA vaccine technology. The presentation will then be concluded with the conclusion and references. Immunization is when the body's immune system becomes resistant against an infectious agent, and one of the most simple and effective way to achieve this is through vaccination. Vaccination is when you introduce a foreign antigen into the body to evoke the body's immune system to respond and fight against the pathogen, thus acquiring adaptive immunity and subsequent protection from the pathogens. Vaccination is considered a health intervention to the public in prevention and treatments against life-threatening diseases, including prevention of spread of infectious diseases. According to the World Health Organization, getting vaccinated prevents 3.5 to 5 million deaths annually, and currently with the rapid development of science and technology, there are more than 20 vaccines available against infectious diseases, which also includes the SARS-CoV-2 virus, which took the world by storm in 2019. Before we continue on, I would like to share some literature reviews describing about the development of using the messenger RNA as a vaccine strategy, which includes the design and manufacturing, the advantages and disadvantages, the challenges and limitations, future innovation for improvement and application in the clinical field. Messenger RNA, or also known as mRNA, is a molecule that contains instructions that can prompt the cell to produce a protein through a process called transcription and translation. Using mRNA as a basis, mRNA-based vaccination works by introducing an exogenous mRNA into the cells, which will instruct the body cells to produce the antigen of an infectious pathogen using the cell natural core machinery. The body will then see these antigens as invaders, which would make the immune system to dispatch immune cells for production of antibodies to fight and clear off the pathogen found in the body, hence training the immune system for any future potential attacks from the same or similar pathogen. In addition to that, more and more people are turning to mRNA vaccine technology due to their innovative and rapid manufacturing process, as which can be found at a scalable rate in response to a global pandemic, unlike traditional vaccines. There is a growing importance in the development of vaccination due to recent pandemics and emerging of new infectious diseases. In the mRNA vaccine technology, the synthetic mRNA structure consists of a 5' cap, 5' UTR, an open reading frame, 3' UTR, and a poly-A tail. Unlike the endogenous mRNA, there has been a few engineered changes in the synthetic mRNA that has been done in order to improve its stabilization, maximize the translation process, and produce strong immunogenicity. In the 5' cap, there is the 7-methylguanosine that is linked together to the 5' end nucleoside of the mRNA through a triphosphate linkage bridge. This additional structure of the 5' cap provides protection to the mRNA from being degraded by exonucleases. Furthermore, it also synergistically works together with the poly-A tail to effectively recruit the ribosomes which are needed for translation. The length of the poly-A tail also contributes to mRNA structure, half-life, and stability. Scientists have discovered that a length of around 100 to 150 base pair is sufficient and required to increase the interaction rate between the poly-A tail and the binding proteins that assist in forming complexes for the initialization of translation. The 5' and 3' UTR untranslated region are vital coding regions which facilitates mRNA translation and also enhances its stability. In addition to that, the UTR sequences were engineered and modified to eliminate the microRNA-induced binding sites and AU-rich regions, which actually promotes the formation of secondary and tertiary structures such as the hairpin. This is important as it helps to improve efficiency of translation and prevents premature termination in the translation process. ORF or the open reading frame are crucial to the mRNA structure as it contains the coding sequences that will be used for translation in the production of the protein. In order to optimize this process, scientists have used the strategy of replacing rarely used codons with frequently used codons that encodes for the same amino acid residues, as this proves to help enhance the efficiency of translation and transcription. Modified nucleosides such as pseudouridine and N1-methyl-pseudouridine are also used to maximize the translation process by preventing the detection of pattern recognition receptors, which induces termination of translation. Together, all of the components in the mRNA structure work together to enhance the translation of antigen protein synthesis, leading to the immobilization of the immune system. mRNA is naturally a large and negatively charged structure which cannot pass through the anionic bilayer of the plasma cell membrane, and in the body, it can easily be targeted by the immune cells as well as metabolically degraded by nucleuses. To address this concern, mRNA delivery vehicles were created and used to increase the efficiency of the mRNA vaccines by transfecting the immune cells without any toxicity or evoking any unwanted immune response. Lipid-based nanoparticles (LNPs) were famously used in the development and deployment of the mRNA vaccines targeting the SARS-CoV-2. It consists of four components, which are the ionizable lipid, cholesterol variants, helper lipid, and the PEG glylated lipid. The LNP is known to have an easy formulation of production, great biocompatibility with the mRNA and human body cells, and a large loading capacity for the mRNA structure in its core, which also provides protection to the mRNA. All four components of the LNP work together by promoting the fusion of the LNP with the endosomal cell membranes, resulting in the release of the mRNA into the cytoplasm of the human body cells. Another mRNA delivery vehicle is the polyplex and polymeric nanoparticles, which are usually consist of cationic polymers condensed nucleosides that can take up various shapes and sizes, which also enables them to be taken up into the cells through endocytosis. However, in comparison to LNPs, they have a lower transfection efficiency, and the production can be more complex and costly. The production of the mRNA formulated into lipid nanoparticle vaccines can be done in a cell-free manner, which firstly starts with the sequencing of the pathogen and identification of the targeted antigen. Then the sequences of the antigen will be designed and inserted into a plasmid DNA construct. The plasmid DNA construct will then transcribe the mRNA by using bacteriophagous polymerases in vitro, which will then be purified using high-performance liquid chromatography to eliminate any contaminants and reactants. The purified mRNA will then be mixed with lipids in a microfluid mixer to form into lipid nanoparticles. Rapid mixing will cause the lipid to encapsulate the mRNA structure instantaneously and precipitate as self-assembled nanoparticles. The nanoparticle solution is then filtered to remove non-aqueous solvents and any encapsulated mRNA. And the filtered mRNA vaccine solution is then stored in sterilized vials, which will be ready to use. The mRNA vaccines are known to provoke an innate and adaptive immunity response, suggesting the production of strong immunogenicity. It does this through the endocytosis of the exogenous mRNA by antigen-representing cells, which are sensed by toll-like receptors 3, 7, 8 in the endosomes, as well as RIG1, NOT2, LGP2, and MDA5 in the cytosol. This will then induce strong inflammatory IFN1 responses, thus triggering pro-inflammatory cytokine production, thereby activating innate immunity as seen on the left of the diagram. On the right, it describes how mRNA vaccine promotes the activation of adaptive immunity, where the mRNA is translated into antigenic protein by the ribosomes, whose presence within the cell will stimulate the immune system. The antigen protein will then be broken down into smaller fragments by the proteasome complex, and its antigens will be displayed on the cell surface towards cytotoxic T cells by major histocompatibility complex 1 MHC class 1 protein. This will actually result in the activation of cytotoxic T cells to kill infected cells through apoptosis. Additionally, the mRNA encoded or re-endocytosed proteins are degraded as peptides will also be presented on the surface of the cells to helper T cells through the MHC class 2 proteins, which will actually promote the activation of phagocytes and B lymphocyte cells to produce neutralizing antibodies in order to clear any circulating pathogens throughout the individual's body. In comparison to traditional vaccines, the mRNA vaccines have the advantages of rapid design and production as they can be manufactured in a vitro cell-free manner with great flexibility as multiple antigens can be encoded into the mRNA for antigenic protein production with development at a scalable rate and can be deployed quickly in response to a global pandemic. Besides that, they are also non-replicative and do not integrate into the host genome as they do not require to cross the nuclear envelope and interact with the genome to function, hence preventing any transcriptional mutagenesis. mRNA vaccinations were fully catalyzed and developed for deployment during the fight against the outbreak of SARS-CoV-2. These vaccines were acknowledged to be developed and produced through a validated platform by Pfizer, BioNTech, and Moderna, which demonstrated high efficacy in combating against the SARS-CoV-2 global pandemic along with its robust safety profiles. The rapid development of the mRNA vaccines from sequencing, identification, and synthesis to authorization have all occurred within a period of 1 year, which highlighted the technology adaptability and scalability. Clinical trials and real-world studies have also confirmed the effectiveness of the mRNA vaccines against SARS-CoV-2 and its variants. At the start of 2021, a year after the first COVID-19 case was reported in China, SARS-CoV-2 have infected more than 150 million people's lives worldwide. Although most SARS-CoV-2 infections are not life-threatening in younger people without pre-existing medical conditions, severe infection can trigger an unchecked immune system response in the lungs that destroy epithelia and alveoli. This damage can actually cause pulmonary edema, leading to dangerous increase in vascular permeability, which may result in death. Most SARS-CoV-2 vaccine candidates induce an immune response to the spike protein on the virus surface. The spike protein binds to its receptors on the host cell surface angiotensin-converting enzyme 2. The attached spike protein is then cut open by the cell's transmembrane serine protease 2, which induces a conformational change that exposes the spike protein's fusion peptide and facilitates fusion with the cell or endosomal membrane. The two lead of mRNA vaccine candidates, BNT162b1 and BNT162b2, uses LNPs that are formulated using Aquatus Therapeutics ionizable lipid and a nucleoside-modified mRNA in which all uridines are replaced by N1-methyl-pseudouridine to enhance mRNA translation. BNT162b1 encodes a trimerized secreted version of the spike protein's receptor binding domain, whereas BNT162b2 encodes the full-length SARS-CoV-2 spike glycoprotein with two proline substitutions in the S2 subunit, which actually helps to lock the protein in its pre-fusion conformation. In preclinical studies in rhizome macaques, 2 doses of BNT162b2 delivered 21 days apart elicited neutralizing antibody titers 18 times higher than convalescent patient serum, as well as strong CD4 and CD8 T cell responses. In phase 1 clinical trials of the two candidates, 2 doses delivered 21 days apart induced high antibody and neutralizing titers and robust CD4 and CD8 response with mild to moderate adverse events. Both candidates were well tolerated and efficacious, but only BNT162b2 vaccine advanced to the phase 2 and 3 trials, owing to its milder systemic and local adverse reactions in comparison to BNT162b1. In phase 3 trials involving around 40,000 participants, BNT162b2 showed 95% overall efficiency at preventing COVID-19 and more than 90% efficacy across subgroups defined by age, sex, ethnicity, baseline body mass index, coexisting conditions as summarized in the figure above. 1 week later, Moderna vaccine mRNA-1273 was also authorized for the use in the USA. Ultimately, they were the first SARS-CoV-2 vaccines to be authorized in the USA, the UK, Canada, and several other countries. Moderna actually developed mRNA-1273 in collaboration with the National Institute for Allergy and Infectious Diseases at the National Institute of Health and the Biomedical Advanced Research and Development Authority. The vaccine uses the ionizable lipid to formulate LNPs that encapsulate an N1-methyl-pseudouridine modified mRNA. The sequence encodes the SARS-CoV-2 spike protein with 2 proline substitutions as well that confers the prefusion conformation. In a preclinical trial, 2 doses of vaccine, a primer dose and a booster injected into muscle-induced robust virus, neutralizing activity as well as potent CD4 and CD8 T-cell responses in mice. It also protected the animals from upper and lower airway SARS-CoV-2 infection for at least 3 months after vaccination. Hence, these encouraging preclinical results extended to clinical trials in which mRNA 1273 was exceptionally efficacious and well tolerated. In phase 3 trials involving around 30,000 volunteers, 2 doses of the vaccine shows 94.1 effective at preventing the onset of COVID-19. Local pain at the site of injection was the most common side effect seen when the clinical trial was ongoing. After the second dose, half of the volunteers, however, reported moderate to severe systemic side effects like fatigue and fever, but resolved within 48 hours. In real-world conditions involving 3,000 healthcare personnel and other essential and frontline workers in the USA, 2 doses of mRNA 1273 or BNT162b2 were shown to have 90% effective against SARS-CoV-2. Although the Pfizer, BioNTech, and Moderna vaccines have demonstrated excellent efficiency and safety, their need for a cold chain storage logistic actually shows difficulty around some of the warmer and low economic countries, as the mRNA 1273 can be stored at 4 to 8 degrees for a month and only 12 hours at room temperature, while BNT162b2 requires storage at around minus 60. Another mRNA vaccine, which is CBN called by CureVac, uses LNPs formulated using an ionizable lipid and unmodified mRNA sequence, which actually encodes the full-length spike protein with the 2 proline substitutions. CureVac candidate vaccine is stable for 3 months at 5 degrees and 24 hours at room temp. In phase 1 clinical trials, a primer and a booster injection of the vaccine were administered 28 days apart, generating a neutralizing antibody similar to those in plasma from patients who were recovering from COVID-19. And this was actually shown to be well tolerated among the candidates. Unfortunately, in phase 2 and 3 trials involving around 40,000 participants and spanning around 10 countries, the vaccine only demonstrated 47% of efficacy against COVID-19 infection. At the present, vaccines for infectious diseases are the most advanced application for mRNA as a prophylaxis and therapeutic strategy. In the diagram as seen above, the mRNA vaccines in development to protect against an array of common pathogens using disease-specific targeting strategies, for an example, like the spike protein of the severe acute respiratory syndrome SARS-CoV-2, the hemagglutinin protein of influenza virus, the membrane and envelope protein of the Zika virus, the fusion protein of respiratory syncytial virus RSV, and surface glycoprotein of human immunodeficiency virus HIV. In addition to that, each pathogen actually poses a unique set of challenges, including high lethality, rapid mutation, immune evasion, along with new strains and variants. Influenza viruses are responsible for an estimated around 650,000 deaths annually worldwide, according to World Health Organization. They are part of the Orthomyxoviridae family and consist of a single-stranded RNA genome. Current vaccines are targeting the hemagglutinin proteins of the virus, which actually facilitates the virus entry into human body cells. However, the virus is known to mutate rapidly and requires an annual review and modification. Conventional influenza vaccines are live vaccines, which are inactivated influenza viruses that are grown in chicken eggs. However, the viruses mutate for optimal growth, which often render them quite ineffective in humans. Hence, there is a real need for an alternative method in order to address the challenging and tedious production of these vaccines. Synthetic mRNAs transcribed in vitro can meet this need and ensure rapid vaccine production in the event that an entirely new influenza strain emerges. There has also been work towards a universal influenza vaccine that will not require yearly modification. Notably, the conserved stock region of the hemagglutinin, which is not prone to mutation, has recently emerged as a novel universal vaccine target since 2016. Moderna has been evaluating 2 influenza vaccine candidates that are mRNA vaccines based, with producing excellent seroconversion and seroprotection with tolerable safety profiles. Zika virus infection was first identified in 1947, and patients infected with Zika are often asymptomatic or experience mild symptoms such as fever, rash, muscle pain. However, Zika emerged as a global health crisis during 2015 to 2016, which causes severe fetal neural malformations and fetal death during pregnancy. It is primarily transmitted through Aedes mosquitoes and is part of the Flaviviridae virus family, consisting of a single-stranded positive sense RNA. Fortunately, all Zika infections are caused by a single serotype, suggesting that vaccinating against an antigen from any of the strain could protect against all Zika strains. The membrane and enveloped protein, PRME, is a common antigen choice for mRNA vaccines against Zika, as neutralizing antibodies against the antigen can prevent viral fusion of the virus. There is also another study that uses a passive immunization approach with the help of mRNA encoding neutralizing ZIKV117 monoclonal antibodies. Interestingly, these results suggest that mRNA therapeutics encoding neutralizing monoclonal antibodies may have both prophylactic and therapeutic activity, suggesting an appealing approach for vaccination towards immunocompromised individuals. One of the leading causes of birth defects and complications in immunocompromised individuals are the cytomegalovirus CMV, which is part of the herpes viridae virus family consisting of a double-stranded DNA genome. Infections may be asymptomatic in healthy people but can be life-threatening for immunocompromised people, with some leading to death. The targeted antigen for the CMV is the presence of the viral envelope glycoprotein GB and GHGL, which is the pentameric complex, which allows the virus entry into the human body cells, followed by subsequently cell-cell infusion events resulting in dissemination of the virus. There has been a few mRNA vaccine candidates undergoing clinical trials that are targeting the CMV, which is done by Moderna, and aims to elicit strong and durable immune responses to prevent infection and transmission. However, up to date, no clinical data has been reported regarding the safety, reactogenicity, and immunogenicity against CMV, despite a few of the mRNA vaccines against CMV are being held in clinical trial. Respiratory syncytial virus, RSV, is the leading cause of severe acute lower respiratory infection globally. According to World Health Organization, it is responsible for an estimated of around 16,000 deaths in children under the age of 5 and more than 14,000 deaths in people over the age of 65. It is part of the Pneumoviridae virus family and consists of a negative sense single-stranded RNA genome. Currently, RSV vaccine candidates focus on targeting the highly conserved F protein, which facilitates the viral fusion. They also use a synthetic sequence mRNA-based vaccine that has been further engineered and optimized to enhance translation and immunogenicity against the virus. Globally, HIV currently affects around 38 million people and is projected to affect up to 42 million people by the end of 2030. The virus is part of the Retoviridae virus family and consists of 2 copies of positive sense single-stranded RNA. HIV infections generally target the vital immune cells, namely T helper cells, which are crucial in immune function response. If left untreated, the virus will further weaken and damage the immune system, leading to a chronic condition called acquired immunodeficiency syndrome, AIDS. Nonetheless, despite decades of research, there are still no vaccine available against the virus until up to date. This is mainly due to the antigenic diversity of the HIV envelope protein and the so-called glycan shield that conceals important protein components of the virus. At the present, there is a mRNA vaccine candidate for phase 1 clinical trial against HIV, which utilizes a prime and boost strategy to prompt targeted B-cell responses with the aim of generating broadly neutralizing antibodies against HIV infection. However, more research is needed for the goal of providing a longer-lasting immune response. Hence, these studies have also turned to the administration of drugs to be used with vaccine for improvement of efficiency against the virus. Currently, there are 2 mRNA vaccine candidates that are targeting Zika virus and HIV respectively, each one in each category. They are still undergoing the early stages of clinical trial. As you can see on the diagram above, there are the clinical trial identifier as well as the phase and status. Then there are also 3 mRNA vaccine candidates targeting the influenza, with 1 mRNA candidate each targeting cytomegalovirus and respiratory syncytial virus. A key challenge for mRNA vaccines is their stability, where they require a cold chain logistic for transport and storage. For storage, the vaccine requires an ultra-low temperature storage of around -60 to -80 to avoid degradation, which remains as a challenge in distribution, especially in low economical and warm climate countries. Another is the manufacturing process, as scaling up the production may require for a more throughput quality check. In addition to that is the public acceptance towards vaccination itself, as there still seems to be some hesitance and misinformation from the public regarding vaccination and their therapeutic properties. mRNA vaccine technology have greatly advanced over the last few decades. The next generation of mRNA vaccines have been targeted to improve stability through improving the efficiency of the delivery system, as this may enable them to be more thermostable in response toward a global pandemic, as a thermostable vaccine is more preferable, especially when distributing towards places with warmer climates. The great flexibility of the mRNA vaccines technology have also encouraged the future production of personalized vaccine, being tailored to an individual immune response profile, as it shows high potential in cancer immunotherapy and other non-infectious diseases, further improving on the address towards the goal of personalized medicine. Overall, mRNA vaccines also managed to transform global health due to the successful achievement with the combat against SARS-CoV-2, which all happened within a year that managed to revolutionize vaccines development and deployment for future response against future global pandemics swiftly. Hence, partnership between governments, academia, and industry of advanced mRNA technology is required for further improvement and enhancement of the technology for their full potential to be realized. In conclusion, mRNA vaccine technology offers a transformative and revolutionary approach to combating emerging infectious diseases, and continued investment in research, infrastructure, and public education is needed to harness the full potential of mRNA vaccine technology. Here are the references that were used for this presentation. That is all from me. Thank you very much.